Yeah, no, it's gonna be this. I just need to see the animation.
All right, let's get started. Welcome to the 2023 AP annual meeting, Frank Brook Lecture. As you know, AP has two state-of-the-art lectures. One is named after Frank Brook. The other one is named after Paul Webster that we had yesterday featuring clinical cancer research, given talk by Mark Spilker, which is very well received. So this year, the Frank Brook Lecture, we are very fortunate and honored to invite Dr. Devna Barsagi to be this year's Frank Brook Lecture. Definitely welcome. I don't think we need any introduction for Devna to this group. She's a leader, she's an educator, she's a researcher, and she's a great administrator and holding many important positions and titles. She's a vice president, senior vice president, and a vice dean for science, and the chief scientific officer at NYU, one of the largest medical institutes in the world. And Daphna has made many seminal contributions to the field of pancreatic cancer. Her first signature study was done when she was a postdoc at Cold Spring Harbor in James Ferromysco's lab. And she found the RAS signaling RAS protein facilitates the cellular uptake of nutrients through what you call macropinocytosis. So many people have been following her work afterwards. She is a leader, she's a world authority on RAS signaling and the oncogene in cancers, especially pancreatic cancer. And she continues to study RAS signaling and develop molecular targeted therapy, especially mutant KRAS in pancreatic cancer particularly. Her recent work on interleukin-1 L-beta suggests for us to better understanding the interaction between tumors, microenvironment, and immune system, we need better models. She has been developing a lot of phenomenal models that everybody in the field is using. So we thank you for that, Daphna. Welcome. Please join me in welcome Dr. Daphna Barsaghi for the Frank Brook Lecture.
Thank you. It's good it's dark because you're not going to see me blushing. First of all, I would like to start off by thanking Min Lee and the leadership of the organization for the invitation to present my work in honor of Frank Brooks. I did a little bit of reading about Frank Brooks, and what came out of it is my realization that his career really embodies all the core values that our community stands for. He was an excellent clinician and researcher, collegial, and perhaps most important, a big supporter and promoter of the next generation of researchers. So I'm really humbled by the task to pay tribute to this amazing legacy. So my presentation today will focus on the immunobiology of pancreatic cancer, and particularly I would like to describe to you our work that relates to our efforts to understand the role of site-specific immune regulatory mechanisms in shaping the immune evasion that is so characteristic of pancreatic tumors. Now, fundamentally, these mechanisms operate at multiple scales, which are illustrated in the next slide. So as you can see, these scales are interconnected, but each scale provides a unique output that then dictate and shape the host tumor immune axis. And so today I would like to share with you two vignettes, examples of our work to try to understand how we have used this framework in order to identify new evasion mechanisms. And perhaps there is going to be time to discuss either at the end of this session or over coffees whether we can use this approach to conceptualize new approaches to devise improvements in current therapies that are meant to improve immune activations. So the first example or story that I want to share with you is very much work in progress. And so I would really like to hear any feedback or suggestion that you might have. It has to do with a very well-controlled Okay, I didn't realize it was not progressing. So we're going to talk about the site-specific regulatory mechanism using the mesosystem as the example, which is the organ environment. So the first example is based on very well-controlled epidemiological studies that was published already some time ago, controlled both for diagnostic bias and disease stage, which showed that the frequency of development of tumors is in the head of the pancreas is significantly higher compared to the tail, and that the 3-year survival rate, when normalized to disease stage, and this is early disease stage, is lower, significantly lower in head tumor compared to same stage tumor in the tail. So on the face of it, what this suggests then is that the environment in the head of the pancreas might be more permissive for tumor growth. And so we wanted to test this premise, and in order to do it, the first thing that we wanted to establish is an experimental system in which these differences could be recapitulated. So this was a work that was initiated by Erica Ma, a graduate student to the lab, and she was joined later on by Anthony Sorrentino, who is a fellow in the lab. Much to their gratification, what they first saw is that in the KC model, these are mice that are 2.5 months old, you could actually observe a marked difference in the development of the pannins and the stromal reaction in the head versus the tail of the pancreas. A similar difference was observed in orthotopic transplantation experiment, where again they saw that the transplant in the tail of the pancreas are lagging behind the head, and this was associated also with improved survival for animals that bear tail tumors compared to head tumors. So this particular approach then led to the hypothesis that these are not cell intrinsic differences that contribute to the permissive environment in the head of the pancreas, but rather it's the environment in the head of the pancreas that is probably promoting to a greater extent the development of the tumors. And that then led to efforts by Erica and Anthony to try to understand the microenvironmental determinants in the head of the pancreas that promote this preferential growth. And so what they did was to profile the immune environment in the head versus tail of the pancreas. They have done it both in mouse and then validated all the critical data in human samples or human data. And just to cut through the chase, and then we'll go back a little bit, what they found out is that the head of the pancreas preferentially display a pronounced Th2 immune response. And this was observed first by the production of Th2 cytokines, predominantly IL-33, which they saw produced abundantly by fibroblasts. TSLP, same differences. And then a whole plethora of cytokine, Th2 cytokine and chemokines. The bar graph are the data from mice, and the dot plots are from human data using TCGA as a database. Now, this was accompanied by the recruitment of Th2 cells, particularly eosinophils. Again, human data, much more abundance of the eosinophils in the head versus the tail, and same hold for the mouse. And perhaps more importantly, when they depleted eosinophils in the mouse, what they found out is that there is a significant reduction in the ability of the head tumors to grow. So essentially, what these suggested then is that the establishment of the tumors in the head of the pancreas is dependent on Th2 immune response that is happening locally in this region of the pancreas. Now, as some of you know, Th2 response has a very important host protective function, predominantly in the context of protecting the organism from extracellular parasites and helminths. It is also involved in regulation of metabolic homeostasis. And last but not least, and probably very relevant to this audience, it is actually important for the orchestration of tissue repair in response to injury. Now, I would say at the outset, we have no idea what is it that results in an enhanced Th2 response in the head of the pancreas. We have some thoughts about it, so again, maybe there is going to be some time to discuss it. But what we do know is that part of the host protective response of Th2 response involves the reduction of tissue inflammation, which is regulated by suppression of type 2 immunity. And since type 1 immunity. And since type 1 immunity is really the major immune axis for anti-tumor effect, we think that perhaps this cross-regulatory network is the one that is responsible for the enhanced growth of the tumor in the head of the pancreas. And so what I would like to leave you with is that by looking at these very site-specific immune modulatory events, we might be able to identify new targeting possibility that in this particular case, perhaps take advantage of these cross-regulatory mechanisms between type 1 and type 2 immunity. So I would like to move now to the second example that I want to share with you. And this is really taking advantage of a process that is called organotropic metastasis. And it refers to the propensity of metastases to target specific organs. And the premise is that this specificity is due to the fact that the cells, the tumor cells that arrive to the metastatic site are able to take advantage of organ-specific immune escape mechanisms. Now, in the case of the pancreas, the two relevant models are, or the two relevant organs are, of course, the most common metastatic site in this disease, which is the lung and the pancreas. And what we have decided to do then is really to try to understand how this principle is applied in the context of these two metastatic sites. And so the first thing was really to, again, establish an appropriate experimental system. And we did it. This is a work that was done by Benny Shapiro, a graduate student in the lab, and Chi Li, a postdoc in the lab. And what they've done was first to isolate tomato lineage labeled KPC cells from animals that were 4.5 to 5 months old, and the cells were isolated from spontaneous metastases that were generated in these animals. Okay, so the first thing that they did was to take these two cell lines from the lung and the liver. They kept them in culture, and they saw that they grow to the same extent. The proliferative rate is identical. But then when they put them to the liver versus the lung cells and the liver cells to the liver, what they found out is that the lung cells are having a very hard time, if at all, to grow in the liver. And so that was an indication that the lung provide the lung metastatic cells with a permissive environment that does not exist in the liver. So to identify the mechanism that provides this organotropism to the lung cells, what we first did was to compare by bulk RNA-seq the organ-specific expression patterns of liver, lung, and liver and lung metastatic cells, and then compared it to human data. And this was the Venn diagram that was generated from these results. And the upshot of it was that the lung metastases cells have a unique signature of 39 genes. And one of the most upregulated genes in this signature was a gene that encodes for the serotonin transporter, or SERT, SLC6-4A. Now, because of the fact that serotonin has been shown to play an important role, particularly recently, both in terms of regulating the phenotype and the function of immune cells, we decided to pursue it further. So the first thing that we did was just to validate the in silico data, and what you can see here, we could observe a very robust upregulation of the serotonin transporter in the cells by qPCR, but also by a functional assay, which involves simply incubating the cells with fluorescent serotonin. And you can see that the lung cells are taking up the serotonin much more robustly than the other cells. We then moved to do the functional assay. And here what we found out is that the knockout of the serotonin in the lung cells was inhibitory to the growth of the lung metastases, but had no effect on the growth of the liver mets. And so, given that the function of this transporter is to transport serotonin, the conclusion that we took away from this selectivity or selective sensitivity to the knockdown of the transporter was there is something about serotonin homeostasis in the lung that is really conferring on these cells' growth advantage. So serotonin is not a common entity in the world of pancreas cancer. So I thought I'll just remind you that it is made through a very simple two-stage enzymatic reaction with the key and rate-limiting step is the first step, which is mediated by TPH. So this is the enzyme. Now, there are two different isoforms of the enzyme, TPH1 and TPH2. TPH1 is the peripheral, the enzyme that is responsible for peripheral production of serotonin, and TPH1 is the central nervous system. The important thing is that it's very difficult to measure serotonin level on tissue, and as a result, TPH1 is used as a surrogate for serotonin production. So what did we see? First, what we found out, even in the lung, this is mouse data, even in the normal lung, there is a robust expression of serotonin based on TPH1 immunohistochemistry in the bronchial airways. And this happens either in the absence of metastases or in the presence of metastases. In the liver, there is no evidence for the production of serotonin, again, both in the normal liver and a liver that bears metastases. The key thing was that the same difference was observed also in human. You can see robust production in the lung and relatively constrained production in the liver. Now, as some of you probably know, the bronchial airways consist of two cell types, the epithelium and the smooth muscle cells. And what we found out is that the serotonin is produced by the smooth muscle cells that surround the bronchial epithelium. Okay, so this then raises the possibility that the serotonin production in the lung might serve, in fact, as the host-dependent organ-specific determinant that allows lung metastatic tropism. And so to understand better this mechanism, the first thing was we looked at the expression pattern of basically bulk expression pattern of the wild type and knockout lung cells. And what we found out was that although there were some differences, genotype-specific differences, when we took the 50 top hits for each genotype and did pathway analysis, we really didn't find any significant differences. So this suggested that there is really no cell-intrinsic mechanisms that are responsible for this serotonin-dependent tropism. And with that, we moved to the immune cells in the lung and their response to changes in serotonin transport. And so the first thing that we did was single-cell RNA-seq between comparing the wild-type and knockout cells. And what we found out was that there was a significant contraction of a very important immune cell population. These are arginase-positive macrophages, interstitial macrophages. So these are the sort of the card-carrying M2 macrophages in the lung. These are the ones that promote tumor growth and immune suppression. This particular population of cell was also enriched for a serotonin-regulated pathway, and then we verified that by flow that there is indeed a contraction of this population. And finally, both by pharmacological and genetic approaches, we were able to show the depletion of these macrophages is responsible for the growth of these lung mets in the presence of a serotonin transporter. So let me just tell you briefly about these particular cells and how they become immune suppressive. These cells are known to become immune suppressive through a process that is called efferocytosis. This is the process by which the cells are taking in or phagocytosing dead tumor cells. And they do it as a way of preventing the inflammatory response that could be caused by apoptotic cells. And again, this is something that is mediated by the production of anti-inflammatory cytokines. Because we have the tumor cells are red, we can see actually the process of efferocytosis in situ. I just showed you 2 examples here. It's very cool. I mean, you can actually see the macrophages in action taking the tumor cells. Now, the key thing is that efferocytosis is inhibited by serotonin, and this inhibitory effect requires the transport through the same transporter. So we have a situation of a transporter on the tumor cells and a transporter on the immune cells. And you can probably see where I'm going to is a tug of war between these 2 cell populations. So what we then decided to do was to treat the animals with fluoxetine so that we inhibit completely the transport of serotonin through the receptor. And what we found out is that under these conditions, there is an increased efferocytosis or uptake, you can see on the top panel, or uptake of the tumor cells, and also increase in the amount of immune suppressive cells of this class. Okay, so this is the last and the summary slide. And I think it really brings home the important point that we all need to think about when we talk about the environment of a certain pathology that we are studying, which is really the spatial relationship between the various players. So think about this as a process. In the normal lung, before the tumor cells have arrived, there is a lot of production of serotonin, as you saw, by these smooth muscle cells. And in essence, this keep these immune suppressive interstitial macrophages at bay in terms of being immune suppressive. With the arrival of the tumor cells, we have a completely different scenario. The tumor cells are now serving as a sink for the serotonin. The macrophages can now start engulfing the tumor cells, and that in turn will create the immune suppressive environment that the lung metastatic cells will require in order to continue and thrive. And I just wanted to add the 2 images that show again the importance of the spatial proximity. So you see in green on the top picture, you see the immune cells, the tumor cells surrounding the airways, which means they're very close to the source of serotonin, so they can really absorb the serotonin that is generated by the airways. And then on the bottom picture, what you can see is the configuration of the airway, the tumor cells, and then proximal to the tumor cells are these immune suppressive macrophages. So I want to leave you with these 2 conclusions that I believe are worth thinking about with respect to most of the things that we study that involve tumor host interactions, and particularly tumor micro and macro environment. The first is that the site-specific tumor cell interaction drive distinct immune landscapes, and they can be completely distinct. So when we think about how to devise strategies that improve immune activation, we need to take it into account. And the next, which is an open question, is if we really devise methodology to identify site-specific nodes of immune modulation, is it really a necessity in order to improve therapeutic success? So I will close here. Happy to address questions now or later. First of all, acknowledging members of my lab. You can see them in the picture. We are a happy group, but also recognizing a lot of collaborators that we have interacted in the context of this work and other pieces of work. As I said, this is a very collegial community, and I think we're all blessed by having each other in order to work together. And of course, last but not least, funding agencies. So thanks again for your attention, and if there is time, I'm going to be happy to take some questions.
Thank you, Devna. Thank you for the outstanding talk. No question. I'm sure Dr. Barsaghi will be available during lunch and coffee break for questions. I'm invited Dr. Saluja to come to the stage to present the Frank Brook Lectureship to Dr. Devna Barsaghi. So the next session will start now. The co-moderator, please come to stage. Yusufumi.